So the next structure we're going to look at is lipopolysaccharide, essential in gram-negative bacteria. So at the base you have this unusual lipid called lipid A, and then you've got a bunch of sugars which form initially an inner core, or the core oligosaccharide here, which is divided up into the inner and the outer core. Now, the inner core, you have a weird sugar, which KDO, I think, stands for keto deoxyoctuloside or something like that. Some kind of weird 8-carbon sugar. You only find it in LPS. And also heptose. Then afterwards, the outer core, variable sugars. Here you've got N-acetyl glucose, glucose galactose. Now the inner core, now let me just get this straight here. So lipopolysaccharide, all gram negatives have got lipid A. That's the same in any type of lipopolysaccharide for any gram negative bacterium. The inner core structure is conserved and identical among bacteria of the same genus and often of the same family. So this is always conserved in all gram negatives. This is always conserved in gram negatives of the same genus or the same family. And the outer core, generally it's identical for different strains of the same species of bacteria. But there are some cases where the outer core, you can have 2, 3, 4, 5 different variants within a single species. And then finally, you've got these O antigen repeats. So whatever it is, the outer or the inner core, it's composed of, 1, 2, 3, 4, 5, 6, 7, 8, about 9, 10 sugar molecules. The O antigen repeats, each repeat is 3 to 8 sugar molecules, and you can have 10 to 40 repeats. Okay, so you've got these short subunits that are repeated in a big, long, linear molecule. And these O antigen repeats are highly variable. You can have tens of different serotypes within the same bacterial species of the O antigen. So basically, this whole structure is becoming less and less conserved the further out you get from the base, okay? This is identical in all gram negatives. This is identical in species within the same genus or family. This is generally the same within a species, and then this is very, very variable. Now, what is the function of this molecule? Well, it's basically there to protect the outer membrane against chemical attack. So, of course, detergents, alcohols would disrupt the outer membrane, and also things like bile salts in our digestive tract. Some antibiotics also attack the outer membrane of gram-negative bacteria, and antimicrobial peptides that we produce also disrupt the outer membrane. And LPS, lipopolysaccharide, gives gram-negative bacteria some protection against this type of chemical attack. Now, because the lipid A is such a highly conserved molecule present in all gram-negatives, we have receptors in our innate immune system that can recognize the lipid A part of LPS. And if these receptors are activated, then they cause inflammation. Okay, so this is why LPS is called endotoxin, because if your blood comes into contact with LPS, then you'll start to get fever, diarrhea, septic shock. Okay, diarrhea, okay, if you've got LPS that penetrates the epithelial cell barrier in your gut, you'll get inflammation, diarrhea. If you get any that's in your blood system, you'll get fever, you'll maybe get septic shock. And it's the LPS which is causing this. And the other thing that's important here is that because these repeated motifs are so variable from one strain of bacterium to the other, they are very useful for identification. So in a serotype, the O antigen stands for the repeats in LPS. So O is basically for O's, okay, they're always sugars. And it's one of the serological characteristics that allow you to identify species of bacteria. Now, quickly for the synthesis of LPS, it's the same kind of problem as the synthesis of peptidoglycan. The bacteria have to produce a complicated polymer that's on the outside of the cell. It's the same problem, and basically bacteria are going to use the same solution. So the core oligosaccharide and the O antigen repeats are synthesized separately. So the core oligosaccharide is synthesized on lipid A, first at the cytoplasmic side of the plasma membrane, then the whole thing is flipped over. And you get a similar process occurring with the synthesis of the O antigen repeats. They're synthesized on the cytoplasmic face of the inner of the plasma membrane and then flipped over. Then you've got a transglycositase, which is going to transfer this subunit onto a chain of the O antigen repeats. And then once the chain has accumulated 10 to 40 repeats, it's going to be transferred onto the end of the core oligosaccharide. And then the last step is you have a big protein complex that crosses, that's present in the plasma membrane, crosses the peptidoglycan layer, and also a component in the outer membrane. It's just going to pick this whole thing up and carry it out to the outer membrane. And this requires ATP, which is consumed inside the cytosol. But because the whole thing is connected, you can perform this, physical work to move the molecule from one place to another. Okay, so that's LPS synthesis. Now, can anybody guess what this is? This lipid here. Bearing in mind that if I think you can guess it, there's probably some information you've already seen that allows you to get the right answer. And the little clue was, I said, the problem for producing LPS is the same kind of problem as for producing peptidoglycan. And to solve the same problem, bacteria just use the same method, same mechanism. Bacteria, just like everybody, they're a bit lazy. They're not going to invent something from scratch if they've got something else that's working. Okay, I'll just use that. It works. Just come on, let's just change it a bit. It'll work. Nobody can guess? No, chose the wrong one. But that UDP will be involved in making this attachment here. Yes. So it's not illustrated on the slide, but it will be in a step just before here. But what is this? What was this before? Because it's the same. Bactoprenol diphosphate. So it's really exactly the same thing that's happening here, okay? So for, yeah? I just didn't really get what bactoprenol is. Okay, in my diagram, I've got bactoprenol. This is the symbol for bactoprenol here, some kind of like vague cylinder. And it's got a phosphate group. So this part is kind of fatty acid chain. Okay, so it's like and this is a phosphate group. So it's a little bit like a lipid, but simplified lipid. Okay, so it's in the plasma membrane and it can flip from one side to the other. But the phosphate group is always, outside the lipid bilayer. Okay, so once again, this is bactoprenol, which is being used to attach the repeating subunit and to flip it over in the periplasm, in the plasma membrane. So same kind of problem, same kind of solution. Okay, now another structure on the outside of some bacteria is the capsule. So as we saw before, you can see it on light microscopy. It's this kind of like diffuse zone around the outside of a bacterial cell. And on electron microscopy, you can sometimes see a bacterial capsule as this kind of quite thick region around here, also called the glycocalyx sometimes. Now, so the capsule is a polymer on the outside of bacteria. Now, the polymer is most frequently polysaccharide, but can be polypeptide. And it's a facultative structure. Not all bacteria have them, but you can find them in gram positives and gram negatives. So not an essential component, can be present or absent. Now, in serology, capsule antigens are called K antigens. So they can be present or absent, and if present, they can have different structures. Okay, because the repeating subunit, once again, can be different between different strains. Now, what is the function of the capsule? Well, once again, it's got a kind of protection against chemical attack. It's a little bit like LPS in that respect, but also it can protect against infection by bacteriophages, particularly in gram-negative bacteria. A lot of bacteriophages will use LPS as the receptor for binding to the cell to infect it. So if you put another layer on top of the LPS, then those viruses, those bacteriophages, won't be able to get in and infect the cell. So it's protection against bacteriophages, bacteriophage being a virus that infects bacteria. Another function is to protect against the bacteria drying out. If bacteria are growing on a surface, sometimes there'll be water available, sometimes it will dry out. And because these are made mostly of polysaccharides, they tend to retain water. So it can help bacteria to survive if the water, dries out. And last but not least, these capsule antigens are important virulence factors in, several pathogenic bacteria, because they can protect the bacterium from being phagocytosed by neutrophils and macrophages, cells of your immune system. So often the most pathogenic strains of bacteria have a capsule because it allows them to resist the immune system. Okay, capsule antigens are polysaccharides, sometimes polypeptides, on the outside of the bacterial cell. It's the same kind of problem as we saw for peptidoglycan and LPS, and it's the same kind of solution. So the repeating subunit is synthesized on the inside in the cytoplasm attached to bactoprenol diphosphate. There's an enzyme which is going to flip it over, and then a transglycosidase which will attach this subunit to the chain that's being elongated. And then finally, there's a big protein complex which grabs hold of the whole polymer and transports it out to the outside. So the biosynthetic pathway is really quite similar to what we saw for LPS. And it's also a little bit similar to what we saw for peptidoglycan. So, once you've understood how peptidoglycan is synthesized, then basically LPS and capsule antigen biosynthesis, you get it for free. 3 for the price of 1. Amazing. Come on, you never get a deal as good as this in the whole of the rest of the semester. Definitely not in biochemistry, I'm telling you that. Okay, so the other structures you get on the outside of bacteria are these filamentous structures. Now, first, we're going to talk about fimbriae and pili. Now, these are two different words for exactly the same structure. And we have these two different terms because basically two different microbiologists describe these structures and publish them pretty much the same time. So fimbriae, I think this is the Latin for like a thread. And this was a Scottish microbiologist from Edinburgh who described these and called them fimbriae. And pili means like hair, like poil. And I think he's an American microbiologist gave them that name. So even up to now, we have these two terms that coexist, but they are the same thing. Now, fimbriae is a bit difficult to pronounce, so now I'm going to just talk about pili, okay? But it's exactly the same. So you can have up to 1,000 of them per bacterial cell, and you can have several different types present on the same cell. They are very narrow, 3 to 10 nanometers in diameter, and they have different functions. Different types of pili have different functions. Many are involved in adhesion and absorption either to an inert surface or to cells of the host for pathogenic bacteria. Some of them can secrete proteins, and some of them can be involved in gliding motility, movement on a solid surface. Now, as well as these normal or regular pili, there are what are called the F pili or conjugation pili. So these are rather fewer in number, a little bit towards the high end of the range for the diameter of pili. And they deserve their own special name because they have a very particular function. They are essential for the conjugation, that is the transfer of genetic material between bacteria. So one of the bacteria, which is F+, will extrude a conjugation pilus, and this is going to go and latch onto a nearby cell, and then so you get the contact, and then this conjugation pilus is going to shorten, and these two bacteria will come together. So it's like throwing out a harpoon and then reeling it in, and then when they're in contact, that's when the DNA exchange occurs. Okay, so this is a very specialized function, so they have their own kind of like name here. Now, there are different types of pili with different types of functions, and as usual, the functions they perform are related to the structure they have and their biosynthetic pathway. So this slide is a little bit complicated, but it's worth trying to think about it because it helps you to understand which types of pili can perform which types of function. Okay. So now they're rather confusingly called type 3, type 4, and CU or curly pili. So you might wonder, hang on, we got type 3, we got type 4, where's type 1 and type 2? And in fact, type 3 and type 4 are called that because they are closely related to the different secretion systems the gram-negative bacteria have. So I think there are about 6 different secretion systems in gram-negative bacteria. And 1, 2, 5, and 6 are not related to pili at all. Okay, but type 3 and type 4 form these filamentous structures on the outside of the bacteria. Okay, so that's where we've got type 3 and type 4. We don't have type 1 or 2. Okay, now let's start on the left-hand side here. So chaperon, usher, pili, and curly pili. Now, their function is only adhesion. Okay, they don't do anything else. They'll bind to some surface or another cell. And the part that is going to be binding is the distal part of the filament. So for the chaperone usher pili, it's an adhesin, whereas for the curly pili, well, it's just a signal subunit. Now, how are these things formed? They're both anchored on the outer membrane. Okay, so they don't start down here in the plasma membrane, they're anchored in the outer membrane. Now for chaperone usher pili, the main filament subunit, so FIM A or PAP A, they are secreted co-translationally into the periplasm. Now, normally these filament proteins would bind to each other, they polymerize. But in order to prevent that happening, but you don't want this thing to polymerize straight away, right? You don't want to form a filament in here. You want the filament to be formed on the outside of the cell. So the first thing that is going to happen is when the filament protein emerges into the periplasm, it binds to a chaperone, this kind of L shape here. So that binds very quickly to the filament monomer, and then it kind of brings it along and binds to this plate at the base of the filament, and then you get the polymerization here. And little by little, the filament's going to extrude from the surface of the bacterial cell. So for the curly pili, it starts off in a similar way. You have the monomer that's translocated into the periplasm. Then, instead of being added to the base, it's going to be exported outside of the bacterial cell, and the polymerization occurs at the top. So this is the big difference between chaperone, usher, and curly pili. One of them starts to polymerize at the base, one of them polymerizes at the top, at the tip. But basically, once the filaments form for both of them, the bacterium can't control anything because it's all happening, you know, on the outside of the cell. Once the filaments form, that's it, it's there. And they can only be involved in adhesion. So they can't be retracted. Okay. Let's do type 3 secretion pili. Okay, so type 3, if we go into type 3 now, what you can see is they're formed with a kind of a very big multi-protein structure that crosses the inner and the outer plasma membrane and crosses the peptidoglycan layer. And what happens is that the filament proteins, they are synthesized in the cytosol, and then ATP hydrolysis is used to transport the filament protein all the way up the inside of the tube here. And it comes out the end and adds on to the distal part of the filament. So this filament is hollow, okay? You've got to have a protein that's transported up the middle and then adds onto the extremity. And that's why the diameter is thicker, okay? About 10 nanometers in diameter here. Now, once again, the polymerization is occurring outside the cell here. So the bacterium can't control it. So once the filament's formed, well, that's it, okay? Can't be retracted. Now, the function of type 3 pili is secretion, okay? Because the filament protein is transported up the inside of the filament, but also different cargo proteins can be transported and secreted outside the cell. So that's the function of type 3 pili, secretion. Type 4 pili. Now, once again, big multi-protein complex, crosses the plasma membrane, crosses the outer membrane, and a filament that goes all the way through. Now, what is different here is that the proteins of the filament, they're added at the base. Once again, it requires ATP hydrolysis, and the filament proteins are added here, so little by little the filament is extruded. Now what's interesting here is that this process is reversible. So the bacteria can control it, okay, so that they can push out a filament and they can put it back in when they want. And that's why these type 4 pili are involved in adhesion and also motility across the surface. And the last one, type 4 secretion pili. It's the same kind of structure as type 4 pili. So the filaments, proteins are added at the base, but this time the filament is hollow and other proteins can also be transported across the plasma and the outer membrane and secreted. Now, type 4 secretion pili seem to be related to these ones, the conjugation pili, because they have the combination of the different characteristics that are required. So the bacteria can extrude them and retract them, and they're hollow so they can transport some kind of cargo on the inside of them. So the conjugation pili are a kind of modified form of type 4 secretion pili. Okay. Cu, curly, adhesion, type 3 secretion, type 4 adhesion, motility, type 4 secretion. Those are different functions. So that's what gram-negative bacteria have. Gram-positive bacteria don't have so many types of pili. They have something similar to the curly type adhesion pili, but they're anchored on the outer face of the peptidoglycan layer. Okay, so they don't have any outer membrane. This curly type pili in gram-positives are attached on the outer face of the peptidoglycan. Okay, so just to illustrate the motility of some gram-negative bacteria using type 4 pili. So what you'll have is that the distal part of the filament will bind to some kind of surface. And if this bacterium is responding to some chemotactic signal, like a high concentration of glucose over here, and it wants to move in this direction, what will happen is that the bacterium will extrude new pili at the front, and it will retract the pili at the back. And then once these ones have made contact with the surface over here, then they'll, okay, first adhere, okay, and then they'll start to retract. And that's going to generate force, which moves the bacterium across the surface. So, you know, it can pull itself along on a surface. Okay, so it's only possible with type 4 pili, because in order for this to happen, then it's got to be possible for the bacterium to control polymerization and depolymerization of the filament. Okay, I'm not going to ask this question because I think otherwise it should be kind of obvious. No, I don't know. Do you want to go for this question or is it too easy? Okay, you're going on the question, I can see. But okay. Oh, I don't have it. Let's just on a show of hands this one, I guess. No, okay, so you can't vote on this one, but you just tell me. Okay, everybody thought of an answer for this? Okay, has anybody not thought of an answer? No hands go up, so I guess everybody thought of an answer. Good. Okay, who says 1? Can't work because they're hollow. Nobody. Who says 2? Can't work because they're in the external membrane. Nobody. Who says 3? because the filament proteins are at the tip. Yeah, you're all right. Yeah, that's right. That's why. So the polymerization is at the tip. You can't control it anymore. Yeah, yeah, it was too easy, wasn't it? Why didn't you just tell me? Yeah, I know. Next time. Okay, last external structure here are flagella. So flagella, you can have one or several per bacterium, and depending on how they're distributed on the bacterial cell, you have these terms which are monolopho or peritrichous. So something to do with where the hair is on the bacterium. So if you've got one flagellum on one tip of the bacterium, that's monotrichous. If you've got a few, but they're all attached at the same end of the cell, that's lophotrichous. And if they're all the way around the bacterium, that's peritrichous. Don't think you really need to remember one example per bacterium. It's not a big deal. Now, the flagellum is once again made up of proteins. So, all the pili, they're protein structures, the same as the flagellum. And what it looks like is this. You've got a lot of proteins that produce a kind of like a axle here and a base plate which is going to rotate. And then after the axle emerges from the external membrane, there's a kind of hook structure which will curve around the filament, and then the main filament, which is about 15 to 20 microns long, it's just going to be floating around outside the bacterial cell. So these are quite big, quite long structures. They're thicker and they're much longer than pili. Now, to get an idea of the length here, 15 to 20 micrometers, that's probably about 5 to 10 times the length of the bacterial cell. Okay, so this is really massive compared to the size of the bacteria. Now, flagella can be present or absent. Not all bacteria of a particular species will have them. And the particular protein which makes up the filament can be variable between different strains of the bacterium. So once again, it's an antigen that's useful for identifying different strains of bacteria. The K antigens, the O antigens, and H antigens are useful for identifying individual strains of the same species of bacteria. So H is for heat, because the flagellum is made up of protein, so it's destroyed by heat, whereas the capsule antigens and the O antigen repeats in LPS, they're resistant to heat, okay, because they're polysaccharides. Their structure isn't denatured. Okay, so that's the structure, and it's used by bacteria to move around in a liquid medium. And the way it does this is that proton motive force is going to be used to turn around this axle. Okay, so if protons translocate from the periplasm to the cytoplasm, that's going to generate energy that's used to turn this thing around. Now, one last thing about this is that, how is the filament formed? So individual filament subunits are translocated up the inside of the filament, and they are added onto the distal end. So this looks like what that we just saw in pili. Type 3 or type 4? It's hollow, but type 3 is also hollow. Let me go back. Oh, what does it look like? This one or that one? Type 3, because the filament protein gets added on the end. So it's exactly the same system for flagella synthesis. So that probably means that the flagellum is basically, it's once again, it's not invented out of nothing. This is like a type 3 secretion system, and they've just like slapped a motor on the base of it, you know. Oh, let's just stick a motor on there. It's going to go round and round. It's going to be great. So, you know, that's where flagella come from. It's like a modified type 3 secretion system. Now, the problem for bacteria is how to move about in a kind of like logical way that makes sense, that is useful in 3 dimensions, just with a flagella rotor. Now, if you or I were trying to design something to move around in 3 dimensions, right, like a submarine, you'd probably want to have, you know, forward, backwards, left and right, and up and down. So you need to have like 3 different types of motors that are controlling things, and you'd be able to move around. But that's not how the flagella rotor works, because it can only have 2 different different statuses. It can either be going around like this, or going around like that, or just doing nothing. Okay, it can do nothing. So somehow bacteria have got to be able to move around in a coordinated way, just with 2 types of motion. It's the flagellum is either rotating clockwise or counterclockwise. And how it works is if the flagellum is rotating counterclockwise. Everybody in French understand what this means? Good. Then the bacterium is going to go forward. When the flagellum is rotating clockwise, it's going to be moving around in a random direction. So instead of having like up and down, left, right, forwards and backwards, you've got forwards and random, right? And then it's going to be the alternation between the 2 is going to be used to give some kind of logical result. So that's what happens with a monotricious flagellated bacteria. Now, with peritricious flagellated bacteria, all the flagella are rotating in the same sense at the same time, okay? So they're not like half of them going clockwise, half of them going anti-clockwise. They're all rotating in the same orientation at the same time. So either they're all going in an anti-clockwise direction and it's going to go forwards, or they're all going to be rotating in a clockwise sense, and then it's going to be going around randomly. So the rule is basically the same, okay? Forwards or random. And alternating between these 2 modes is going to help bacteria move towards things that they like, for example, glucose, and move away from things that they don't like, perhaps acidic conditions or high concentration of toxic metal ions and things like that. So imagine that you are some kind of bacterium which is here, and initially what you're going to do is have a clockwise flagella rotation for a random time before switching to counterclockwise. So that means the direction is going to change randomly for a while, then it's going to stop, and then for a while it's going to start moving forward. So you've got a random change in direction, and then the flagellum is going to start rotating in an anti-clockwise direction. Then this anti-clockwise rotation is going to be maintained as long as the bacterium is moving up the concentration gradient. That is, as long as it's moving towards the glucose. However, because the initial choice was totally random, the choice of direction was initially random, you're never going to be right on target, okay? You're going to miss the target and start moving away from the glucose. Now, as soon as that happens, what's going to occur is that the bacterium is going to switch the rotation of the flagellum. So it's going to stop moving away and make another random switch. So then it will start to rotate randomly again, and then stop, and then after a random time will start moving in a straight line again. And then little by little, these switches, let's go back for this. So this alternation between random movement and moving in a straight line will mean that the bacterium is always going to kind of move around like this, but it's never going to get very far away from the maximum concentration of glucose. So just using a simple algorithm and switching between random and linear movement, you can get something that is actually useful for the bacteria. Okay, now this is an important question, okay? Which surface antigens are useful for identification? Very, very important to remember this. Okay, mostly correct. Okay, so two highest scoring answers are both correct. So LPSO repeats, yeah, that's the O antigen. Capsule antigen, yes, that's the K antigen. Peptidoglycan, not useful. It's too highly conserved. Okay, it's pretty much the same through all bacteria with some slight differences between gram-positive and gram-negative, but not useful, too highly conserved. And that's the same thing for the core oligosaccharide. It's slightly variable, but generally speaking, it's too highly conserved to be useful to discriminate between different strains of the same species of bacterium. So not so useful, not too highly conserved. The Cu pili, not useful, but almost for the opposite reason. So for the capsule antigen, O antigen and flagellar antigen, if you've got a bacterium that has the capsule, it will have one type of antigen, okay? If you've got LPSO repeats, it will be one type of repeat on the same bacterial cell. But Cu pili, one bacterial cell may have two or more different types, okay? And then they can be different between different strains, but the whole thing becomes far too complicated because for the capsule antigen and the O antigen, each individual bacterium, they've just got one. Whereas for the Cu pili, it can be one, it can be two, it can be several. So it's too complicated to be useful for identification. Okay. So you understand what I mean here. Okay. So if you've got a bacterium that is serotype O157, it can't be O100. But if you had different serotypes of Cu pili, it could be Cu5 and it could also be Cu7 and Cu11. So that makes the whole thing far too complicated to be useful. Okay, so that's about it for the external structures, I think. So now we're coming on to the internal structures of bacterial cells, which you'll be relieved to learn are pretty simple. So some bacteria contain inclusion bodies in their cytoplasm, and their function is just to store nutrients. So several bacteria can produce inclusion bodies to store organic material, glycogen or polyhydroxybutyrate. So these are storing carbon. Many bacteria form granules of cyanophycin, So this is a polymer of arginine and aspartate. So it's a store of organic carbon and nitrogen. And so these are very, very frequent in many, many different types of bacteria. Now, several bacterial types can also store granules of phosphate. So phosphorus can be a limiting oligoelement for bacterial growth. And in that case, if bacteria are growing in conditions where there is a lot of phosphate available, then they'll tend to accumulate it and store it in these granules. Because once the phosphate is used up, if it's all gone, they can't grow anymore, okay? So if they've got a bit of a store when they've had lots of it, then they can carry on growing and use up what they've saved. Now, some bacteria, so that have metabolism based on the oxidation of sulfur, can store granules of sulfur with the same kind of idea that if they're in a situation where reduced sulfur is abundant, they can take it up, store it, and then if it's all been exhausted in the medium outside the bacterium, they can still grow because they've stored it up somewhere. Okay, so inclusion bodies, storage function, inorganic and organic nutrients. Now, most of the time, these granules are just polymers that are in direct contact with the bacterial cytosol, but some of them have a kind of a cage associated with lipid and protein around them. That's the case for beta hydroxybutyrate because this is very, very hydrophobic. So there's a kind of like a lipid protein cage around the granule, and it's also true for sulfur granules. But polyphosphate, cyanophycin, and glycogen, they're just polymers directly in contact with the bacterial cytosol. Okay, the nucleoid, what else to say? What to say about the nucleoid? Well, you can see it on electron microscopy. Okay, so you've got two bacterial cell dividing here. Here's the DNA that's concentrated in the middle of the cell. Now, although bacteria or eubacteria do not have histones, the DNA in the nucleoid is compacted because if you get a bacterial cell and you degrade the peptidoglycan a little bit and you digest all the proteins in the cell to liberate free DNA, you can see that this DNA fiber, the length of it is much bigger than the size of the bacterial cell. So in order to pack all this DNA in there, it's got to be associated with some basic proteins, which are basic proteins or small molecules with a positive charge. Okay, so in bacteria, DNA is compacted with polyamines, not histone proteins. So this will compact the DNA, and they also have proteins called HU proteins, which introduce curves into these filaments of compacted DNA. So, and the combination of these two things is what allows bacteria to pack all of this huge DNA molecule into a quite small cell. Now, another thing that is actually kind of important, and I'll draw this actually, is that inside the bacterial cell, each chromosome has got one origin of replication. And this bacterial origin of replication is attached to the plasma membrane and the peptidoglycan by some kind of anchoring proteins. And we'll see just in a few slides that this is very important for chromosomal segregation in bacteria. Okay. So there are some proteins that attach the origin of replication to the bacterial cell wall. Now, exceptions are archaea. So some groups have got histone proteins just like us. And there are some very weird bacteria called planktomycetes, well, some type of planktomycetes, which seem to have a kind of like membrane structure which is around the DNA. So this was a very, very unexpected finding in 2005. Some bacteria seem to have a kind of simplified form of the nucleus, which they shouldn't really have. So that's kind of interesting. Bacterial cytoskeleton. Now, for a long time, let's say up until about, I don't know, the 1990s, it was thought that bacteria did not have any equivalent to actin or tubulin, the main proteins of the cytoskeleton in eukaryotes. And in fact, there is no real sequence homology, but if you look at the structures, the three-dimensional structures of actin and tubulin, and compare them to the structures of some bacterial proteins, then it was possible to find structural homologues. So the MREB protein, which is in gram negatives, and the MBL protein, which is in gram positives, are the bacterial homologues of actin. And just like actin, it can be found as a kind of soluble monomer, or they can polymerize to form filaments. So here you've got a fluorescence microscopy image of a fusion protein between MBL and the green fluorescent protein in Bacillus subtilis, which is a gram-positive rod. And you can see that the MBL protein forms a kind of like helical filament inside the bacterial cell. Now, the function of MREB and MBL is to force the bacterial cell to elongate. Okay, so cookie have lost MBL. Gram-positive cookie, they've lost the MBL protein. So if you don't have MBL or MREB, the cell becomes spherical. There's nothing to put it into an elongated shape. So the basic difference in bacterial structure or shape, rods or cookie, is just down to one protein, MREB and MBL. Now, FTSZ, this is the bacterial homologue of tubulin, and it plays a very important role in bacterial cell division and chromosome segregation. So, like I said, at the beginning of cell growth, the origin of replication is attached to the cell wall. So what's going to happen is DNA replication is coordinated with cell growth and elongation. So as the DNA starts to replicate at the origin of replication, you'll have one copy of the origin which migrates to this end of the cell, and the other newly synthesized copy of the whole chromosome will also be attached to the cell wall, but it will be migrating to the other side because the cell's growing here, right? And the polymerization of peptidoglycan is here. So the cell's getting longer and longer from the middle. So these two copies of DNA are going to be pulled apart. So that's kind of what's happening here in the slide. So you've got the origin of replication there. DNA replication starts, and the two copies of the chromosome migrate to different ends of the cell. And what happens is that at the center of the cell, you have FTSZ, which starts to polymerize. So at the beginning of DNA replication, FTSZ is in a monomeric form. During replication of DNA, it's going to form a ring. And this ring is what's going to be the scaffold for all the proteins that will pull the bacterium into two different daughter cells. Okay, so the FTSZ protein itself, that's not the protein that's like cutting bacteria in half, but it's the scaffold where all these other proteins come to start dividing the bacterium into two cells. Now that's really kind of interesting because, how much time have I got? Yeah, I'm good for time. The closest eukaryotic homologue of FTSZ is gamma tubulin. So gamma tubulin in mitosis forms a ring, and it's this ring which forms the start of the formation of the mitotic spindle, which is what's going to control where chromosomes go in mitosis. Now, chromosome segregation in bacteria and in eukaryotic cells is very, very different process. It's very, very different. But at the base of it, you have the formation of a polymer of the same kind of protein. So this is kind of like a little clue or a suggestion that maybe at the beginning, the basic players in this process of segregating chromosomes were kind of similar in bacteria and the ancestral eukaryotic cells. So that's kind of, this is too close to be a coincidence, I think, So it's really interesting. Okay, so the last thing I'm going to talk about, and then probably we'll call it a day because we'll have finished with bacterial structures, bacterial endospores. Yeah, and we might even finish early. So bacterial endospores are very resistant structures that allow bacteria to survive for a long time in very unfavorable conditions. They are highly resistant to heat, to desiccation, and to chemical attack. So they're very, very difficult to sterilize. Only gram-positive rods can form endospores, and not all of them. Okay, so it's only some gram-positive bacilli that form endospores. No gram-negatives, no gram-positive cookie. And the ability to form endospores is a kind of element of identification because some families, some genera of bacteria form endospores, others don't. And the position of the endospore can also give you a bit of an idea. So Clostridium and Bacillus are two genera of gram-positive rods. And the position of the endospore is rather different. So in Bacillus it's more central, and in Clostridium it's subterminal. Now I wouldn't really like to identify Clostridium tetani compared to some other Clostridium just because of the difference between this and that, but spore position can be used to distinguish Bacillus from Clostridium. Clostridium are all anaerobes and most Bacillus are aerobes as well, so that's a big difference. Okay, so what are endospores made of and why are they so tough? So you can kind of work from the outside in here. So there's often a loose coat of glycoprotein, the exosporium, and then a very, very thick layer of proteins, external and the internal coat. And then inside you have the cortex, which is a very thick layer of peptidoglycan, which contains a large amount of this small molecule called dipicolinate associated with calcium. And the inside of the cell, or the inside of the spore, is a kind of tiny, metabolically inactive cell. So it's got DNA, it's got the chromosome in there, it's got some ribosomes, so it's got ribosomal RNA, it's got transfer RNA, I believe, and it's got some DNA repair enzymes. And these things, small acid-soluble proteins. So these proteins are associated with the DNA and bind to it very tightly, making it more condensed than in a normal cell. So these endospores are metabolically inactive and they can remain viable for years, okay? Yeah, maybe hundreds of years. Now, it's okay if, the spore coat, the protein here, this is what's going to protect the bacterium from chemical attack. So if you add some detergents on here or some bleach or something like that, it's just going to be absorbed by the spore coat. All these proteins will chemically react with it and nothing is going to get into the inside of the endospore. Same kind of thing with the peptidoglycan, but the dipicolinate has an important role in reactivation, germination of the spore. Now, the other thing is that because this protein coat is very, very thick, it's almost impermeable to water. Okay, not much water can get in here. Now, but a little bit can penetrate. So if you have a, I don't know, okay, so let's think about this. I don't know, I often wonder about what's the best way to present this, if it's the best to present spore germination first or spore formation. So what's the function of the spore? The function of the endospore is to keep the bacterial cell alive in unfavorable conditions. Okay, so you form endospores when everything is bad, there's nothing left to eat, the whole medium dries out, it was going to be a dead zone, so you become dormant and become a spore. So that's okay. This whole thing is very resistant, but at some point, there's no point in being resistant and staying asleep forever, because that's the same thing as being dead. Every endospore has got to be able to wake up at some point. And reactivation, germination of the endospore is an important event. And it occurs often when the endospore is put back in a medium with liquid water where there are some nutrients available, like amino acids or sugars, and heating can also reactivate the endospore a little bit, because that can loosen up a little bit of this protein structure and allow some water to diffuse in. And if you've got the presence of some amino acids, some sugars, they can diffuse into the cortex. Now, by a mechanism which I must admit is rather unknown to me, once these activators get into the cortex, this is going to allow the release of the dipicolinate, which will diffuse outwards. And then it will bind to and activate enzymes in the spore coat, which degrade the protein and allow more water and more nutrients to rehydrate the cell here. And then it's going to start to grow and escape from the endospore. So spores are very stable, but they can be reactivated. And the thing that leads to their reactivation is when the spore is present in a favorable medium. It's got liquid water and it's got some kind of nutrients available. Okay, so that's activation of the spore. What about formation of the endospore? Now, one of the things that is weird here is that you've got a lot of structures on the outside of the endospore, and the endospore is entirely metabolically inactive. So this spore here, there's no way that it can synthesize all this stuff, right? So what is happening is that spore formation occurs during bacterial division. So normally during bacterial growth, one bacterium will grow and produce two daughter cells that are absolutely identical, and they both carry on growing. However, during spore formation, what happens is that you have a cell division which is not symmetrical. You'll get one daughter cell which is smaller and the other one which is bigger. So the smaller one is going to become the endospore, and the larger daughter cell is going to become the sporangium. That is the structure that forms the spore. So what will happen is that the smaller cell will be engulfed by the larger one. Then the smaller cell that's now inside the larger one will shut down its metabolism, just express these small acid-soluble proteins, express some DNA repair enzymes, and then shut down its metabolism. Whereas the larger cell will start secreting the different layers of the spore coat. First the peptidoglycan, which will form the cortex, then the proteins of the spore coat, then the exosporium. So all the biosynthesis work is performed by the larger daughter cell, the sporangium. The endospore itself doesn't synthesize any of the endospore coat. So that's how it works. And at the end, what you have is that the sporangium will actually commit suicide. It's going to kill itself. The larger cell will kill itself, and the only thing that survives is the endospore. Okay, so two things to think about here. What induces sporulation in endospore-forming bacteria? It's generally poor conditions for growth. So when nutrients are exhausted, bacteria can't carry on growing. So this will often induce sporulation. And you can sometimes see that, and we saw some in last year's TP actually, because when bacteria are growing in a colony locally, they can exhaust all the available nutrients, and you can see some gram-positives with endospores inside them in TP sometimes because they've used up all the nutrients. So they form endospores and become dormant. And some spore-forming bacteria, or many of them actually, are obligate anaerobes. So if they're exposed to oxygen, then this is something that's toxic for them. They can't survive. So this can also induce sporulation. And that's definitely the case for Bacillus anthracis, the bacterium that causes anthrax, la maladie du charbon. So it's an obligate anaerobe, and it can kill cattle. And once these cattle are ill, they'll start excreting, vomiting, and expel bacteria in bloody diarrhea. So the bacteria, the pathogens that are excreted from the dying animal, will find themselves outside of the infected animal in the presence of oxygen. So they're obligate pathogens. They can't survive outside of an infected animal. So if they're in the presence of oxygen, this is a message to the bacterium, we're not in a host anymore, can't live. So they sporulate in contact with oxygen and then produce these spores that are very resistant, that can remain in a field for years or decades and still reinfect animals years and years later. So that's why we still have sporadic cases of anthrax in some regions of France and around the world, is because the ground in the field has been contaminated with these resistant endospores. And then grazing animals can sometimes eat them and get reinfected. So sporulation induced by unfavorable growth conditions, nutrient exhaustion, presence of something that's toxic like oxygen for anaerobes. That's the first thing. The second thing is, the interesting thing about this is that, as I said before, normally when you think about bacterial growth, you're talking about one cell that grows, divides into two cells, and they're identical. And that means that there aren't really two different cell types in bacteria. There's no differentiation. Okay, so whereas that's one of the big things that differentiates prokaryotes from eukaryotes. We're complex organisms because we've got maybe a couple of hundred of distinct cell types that make up our body with specialized functions. So we can have a big, complicated, multicellular organism. So bacteria never manage to get that far. Okay, they're mostly unicellular, or maybe they grow in filaments or something like that. But here you can see that you've got two of the basic processes that are required to make a complex organism already exist in bacteria. One of them, cell differentiation. So you start with one parental cell here, and it's going to produce two daughter cells that have very different cell fates. One of them is going to become an endospore and will survive, and one of them is going to produce all the layers of the spore coat and then will die. Two different pathways of differentiation. The other thing that's interesting here is one of these cells undergoes programmed cell death, which is also a fundamental process in differentiation in complex organisms. So these two things, differentiation and programmed cell death, they already exist in a very, very simplified way in some bacteria. So, eukaryotes, we didn't invent something out of nothing here. We probably took something that already existed and just made it way more complicated. Okay, so I think that's probably a good place for me to stop. And we'll talk about that at the beginning when we see each other. What is it, Thursday afternoon? I think so. Okay, so that's it for today, except for Microbe of the Week. So 7 p.m., I'll send out the Microbe of the Week question, and I'll try to answer you if I don't feel too tired and if I don't have to wash the dishes or something. Okay, goodbye everybody.